Hello, this is Eric Strong again, and this is Lecture 1 in this course on antibiotics. The topic of this lecture is the classification of bacteria. The learning objective of this lecture is very straightforward. By the end of the lecture, you should be able to classify common bacteria by gram stain, morphology, and a variety of other discriminating characteristics. When an infectious disease is first suspected, before an appropriate antibiotic regimen can be chosen, one must first hypothesize as to the likely type of infection as well as the most likely responsible pathogens. A prerequisite for understanding bacterial infections and for speaking intelligently about them to colleagues is a basic understanding of how bacteria are named and classified. Most pathologic bacterial species are colloquially referred to by genus name only. For example, Klebsiella, Proteus, Salmonella, and Pseudomonas. Some are referred to by specific species name, as in E. coli, and some are usually referred to by genus name only, but probably should be referred to more specifically, as in Enterococcus, Staph, and Strep. Lack of specificity with these genera are problematic as the pathologic species within them don't necessarily behave the same way or share similar antibiotic sensitivities. When bacteria are classified, there are many different considerations. First, morphology. Are the bacteria cocci or rods, or halfway in between so-called coccobacilli? Second, does the gram stain show them to be gram positive, gram negative, gram variable, or just not staining? Next, what are their growth requirements? For example, do they need oxygen? What types of biochemical reactions are they able to undergo? Some bacteria, specifically Streptococcus, are classified according to serotype. Within a single species of bacteria, further distinction can be made based upon antibiotic sensitivity. For example, methicillin sensitive versus methicillin resistant strains of Staph aureus. And finally, the most recent development in bacterial classification is rRNA sequence analysis, which has helped greatly with formal taxonomy, but which has no routine clinical application. Along with gram staining, general morphology is one of the two most important methods of differentiating bacteria. On the most basic level, there are two bacterial shapes. The first is the coccus, or cocci in the plural form. A coccus is spherical. The other shape is colloquially referred to as a rod, which is generally elongated with straight sides. Occasionally, this shape will be referred to as a bacillus. Unfortunately, bacillus is also a specific genus of bacteria, and using the term to describe the shape can potentially be confusing, so most people generally prefer the term rod. Let's talk about gram staining for a few minutes. The gram stain depends upon the fact that different types of bacteria have different cell wall structures. Most bacteria fall into one of two categories. In the first, the outer boundary of the cell is defined by a thick wall of peptidoglycan, which is a polymer of sugars and amino acid. Just inside the peptidoglycan is a thin plasma membrane, which is, as most plasma membranes, is composed predominantly of a phospholipid bilayer. Bacteria which have this structure are referred to as gram-positive. In the other category of bacteria, the outermost layer is referred to as the outer membrane, which is made up of lipopolysaccharide and protein. Inside of this is a thin peptidoglycan layer. Finally, the innermost layer is the same plasma membrane. These bacteria are known as gram-negative. Gram staining uses these differences to create distinguishing colors after application of a sequence of stains. Let's go over the specific sequence of steps in a gram stain, starting with an unstained slide of bacteria, which is a mix of unknown rods and unknown diplococci, that is, cocci that occurs in pairs. The first step is to apply a dye known as crystal violet, which enters and stains all bacteria purple. Next, iodine is added. Iodine forms a complex with the crystal violet that partially traps it within the peptidoglycan layer. Then the cells are washed with ethyl alcohol or sometimes acetone. In gram-negative cells, the alcohol or acetone destroys the outer liposaccharide membrane and washes out the crystal violet iodine complexes so that these cells become unstained again. However, the crystal violet iodine complexes cannot be washed out of the much thicker peptidoglycan layer in gram-positive cells, so they stay purple. Finally, a counterstain, usually a compound called safranin, is added, which doesn't disrupt the gram-positive cells which have retained crystal violet, but which do stain the gram-negative cells pink. Thus, in this example, the unknown diplococci are gram-positive, and the unknown rods are gram-negative. The combination of morphology and gram stain results in 4 major categories of bacteria. First are the gram-positive cocci. Some gram-positive cocci tend to form clusters of bacteria. This is predominantly seen with the genus Staphylococcus, while other gram-positive cocci form either short chains or pairs. This is seen with Streptococcus, Enterococcus, and Peptostreptococcus. Another category of bacteria are the gram-negative rods. There are many specific bacterial species in here, most notably E. coli and the genera Proteus, Klebsiella, Serratia, Enterobacter, Shigella, Salmonella, Yersinia, Pseudomonas, Stenotrophomonas, and Bacteroides. These are the 2 largest general categories of bacteria. However, there are also the gram-negative cocci. The 2 important entries here are the genera Neisseria and Moraxella. And finally, clinically relevant gram-positive rods are relatively uncommon, but include Clostridium and Actinomyces. You may have noticed that some of these bacteria are written in italics. Those are anaerobic bacteria, that is, bacteria that only thrive in the absence of oxygen. While most anaerobic bacteria can be gram-stained, during clinical discussions they are generally grouped together under the term anaerobes and are not referred to by morphology or gram-staining properties. There are a few bacteria that don't clearly conform to these 4 categories. This is partly because they may have shapes that are either variable or fall somewhere in between cocci and rods and are therefore known as cocobacilli. An example of a gram-positive cocobacillus is Listeria, which can cause meningitis in infants, the elderly, and the immunosuppressed. Gram-negative cocobacilli include Haemophilus, Legionella, both of which are major causes of community-acquired pneumonia, and Bordetella, one species of which causes pertussis, also known as whooping cough. Finally, one particularly nasty bacteria is the gram-variable Cocobacillus acinetobacter, the morphology of which is sometimes listed as rods and sometimes listed as pleomorphic. The term gram-variable is itself defined variably in the literature, but generally means that a bacterial species can stain either gram-positive or gram-negative, sometimes mixed even on the same slide. In other words, Acinetobacter can look like just about anything, which seems fitting given the difficulty treating this frequently multi-drug resistant cause of ventilator-associated pneumonia. There are a large number of additional tests used in complex algorithms to further distinguish clinically relevant bacterial species. Luckily, when it comes to acute bacterial infections in humans, there are only 4 tests that are referred to outside of the microbiology lab. They are the Coagulase Test, the Hemolysis Test, Lancefield Serotyping, and the ability of some bacteria to ferment lactose. I'll talk briefly about each. Coagulase is an enzyme produced by some bacteria that converts fibrinogen to fibrin. Its presence is used predominantly to differentiate the different species of Staphylococcus. Staph species which express coagulase are Staph aureus and some strains of Staph intermedius. Most other Staph species do not express coagulase. This is a particularly helpful distinction since Staph species that are coagulase negative are generally not pathologic and are frequently contaminants when grown in culture. This issue will be discussed a little more in the next lecture. The Hemolysis Test. Streptococcal species are further classified based on their ability to hemolyze red blood cells. This ability is usually tested by culturing known streptococcal bacteria on a media called blood agar, which generally contains either sheep or horse blood. The pattern of growth on blood agar can place a streptococcal species into one of three categories. Alpha hemolytic strep oxidizes the iron within the hemoglobin molecule to create methemoglobin, which results in a green or occasionally brown discoloration surrounding the colonies on the agar. Although this is sometimes referred to as, quote, partial hemolysis, the red blood cells are in fact still intact. Species which fall into this category are Strep pneumoniae and a pseudotaxonomic category called viridin strep. Beta hemolytic strep causes complete hemolysis, which results in a clearing of blood surrounding bacterial colonies. This is seen in Strep pyogenes and Strep agalactiae. Finally, gamma hemolytic strep induces no hemolysis at all. This is seen with Strep bovis and with the genus Enterococcus, which had been considered to be a type of Streptococcus until the 1980s. Lancefield Serotypes are also sometimes used to distinguish species of Streptococcus and Enterococcus from one another. These serotypes are based on specific carbohydrates present on the bacterial wall. Group A serotype includes Strep pyogenes. Group B serotype includes Strep agalactiae. Group D includes Enterococcus and Strep bovis. And Strep pneumoniae and viridins have no Lancefield antigens to categorize. Here are the relationships between Lancefield serotypes and the hemolysis test. If you are wondering what happened to group C strep, not only is there a group C, but there are also groups E, F, G, and H as well. Luckily, none of their members are commonly associated with human disease, so unless you are a veterinarian, there is no need to remember them. The final important microtest I'll discuss today is lactose fermentation. The ability to ferment lactose is a major differentiating characteristic of gram-negative rods. It is usually tested by culturing the unknown bacteria on a media named MacConkey agar. This is actually a cool test, so I'll talk about it just a little bit more than the others. MacConkey agar has 6 critical components. First is the agar, which provides structural support for bacterial growth. There are bile salts and crystal violet, both of which inhibit the growth of gram-positive bacteria. Lactose is present, which can be used as food for lactose fermenting bacteria. This results in acidic byproducts and a decrease in the pH of the agar. Peptides are present, which is used as food for the non-lactose fermenting bacteria. The ammonia that is the metabolic byproduct results in an increase in pH. Finally, there is a dye called neutral red, which changes to a red color at lower pH. Here's what is seen when bacteria are grown on MacConkey agar plates. Lactose fermenters, also known as lactose-positive bacteria, are dyed red and are surrounded by a red halo in the agar. The major lactose-positive organisms are E. coli, Klebsiella, and Enterobacter. Lactose non-fermenters, that is lactose-negative bacteria, appear translucent. These include almost all other gram-negative rods, except for those in a final category known as the lactose slow fermenters, which on the agar may appear red or pink, but without the distinctive halo of the lactose-positive bacteria. There are 2 commonly cited members of this category, Citrobacter and Serratia. I'd like to talk just for a minute more about Enterococcus. As I already mentioned, it was formerly known as Group D Streptococcus until the 1980s. And as I mentioned near the very beginning of the lecture, many people refer to all Enterococcal species simply by the genus name. In fact, some microlabs don't even routinely differentiate Enterococcus species. This is really problematic because these species don't act the same. There are 2 major pathogenic enterococcal species. First, E. faecalis causes the majority of enterococcal infections. We won't discuss individual antibiotics until Lecture 3, but be aware that E. faecalis is relatively less resistant to antibiotics. Of particular concern is ampicillin, vancomycin, and the aminoglycosides. E. faecium causes a sizable minority of enterococcal infections, particularly nosocomial ones. E. faecium is relatively more resistant to these antibiotics. So the bottom line is, if you are concerned about the possibility of an enterococcal infection, it's best to pay attention to the individual species, as it will influence decisions about antibiotic therapy while the sensitivities are still pending from the microlab. Most of the bacteria I focused on so far are the so-called typical bacteria. There are two more brief categories to discuss. First are the previously mentioned anaerobes. Once again, this term refers to all bacteria which grow and reproduce only in the absence of oxygen. They are predominantly found in the GI tract, including the oral cavity. While they are generally stainable via gram stain, they are rarely referred to as gram-positive or negative in clinical practice. Common examples of anaerobes are genera Clostridium, Bacteroides, Peptostreptococci, and Actinomyces. Finally are the so-called atypical bacteria. This is an inexact term applied to bacteria which are particularly unusual in some way or another, usually in cell structure, morphology, biochemistry, and/or life cycle. Routinely cited examples of atypical bacteria are Mycoplasma, which lack a cell wall and are highly pleomorphic; Chlamydia, which lack peptidoglycan in the cell wall, have an unusual multi-phased life cycle, and are obligate intracellular parasites, that is, they are incapable of producing their own ATP; Rickettsia, which are also obligate intracellular parasites and generally require arthropod vectors; and finally Legionella, which, as like the above, does not typically grow on traditional media. Although Legionella is not universally included in the atypical category, since its most common manifestation is an atypical pneumonia, and its preferred antibiotic treatment is similar to other atypicals, it seems to make the most sense to include it here. This has been a relatively long lecture with plenty of unusual names and unfamiliar items thrown about. Don't worry, it will require a lot of experience, either clinical or in the microlab, before all of this becomes second nature. However, if you remember nothing else from this lecture, make it the following chart, which will summarize all of the preceding information into a practical classification of bacteria. Gram-positive bacteria can be cocci or coccobacilli. While gram-positive rods exist, they are typically anaerobic and will be saved for later. Under coccobacilli is Listeria. For gram-positive cocci, if they grow in clusters, they are probably staph and should be further differentiated using the Coagulase Test. If they grow in pairs or chains, they are likely either strep or Enterococcus and should undergo the Hemolysis Test to be further speciated. They can also be further distinguished using Lancefield Serotyping. Clinically relevant gram-negative bacteria can be either cocci, rods, or coccobacilli. Gram-negative cocci include Neisseria and Moraxella. Coccobacilli include Haemophilus and Bordetella. Gram-negative rods should be tested for the ability to ferment lactose. Lactose fermenting bacteria include E. coli, Klebsiella, and Enterobacter. Slow fermenters include Serratia and Citrobacter. Lactose non-fermenters include all other gram-negative rods, such as Pseudomonas, Proteus, and Stenotrophomonas. Anaerobic bacteria include Clostridium, Bacteroides, Peptostreptococci, and Actinomyces. Atypical bacteria include Mycoplasma, Chlamydia, Rickettsia, and Legionella. And the last remaining category is for the gram-variable Coccobacillus acinetobacter. I started this lecture with a brief word about how people commonly referred to specific bacteria. I'll end the lecture by revisiting this issue with some more bacteria shorthand with which one should be familiar. Streptococcus pneumoniae is often referred to as pneumococcus. Haemophilus influenzae is almost always called H. flu. The large family of gram-negative bacteria, called Enterobacteriaceae, is often referred to as enteric gram-negative rods or just plain enterics. This shorthand is potentially confusing since there are many gram-negative rods which are commonly found within the GI tract, which are not formally members of this family. Clostridium difficile is C. diff. Any and all anaerobic species are usually called just plain anaerobes and are rarely referred to as individual members, with the exception of C. diff. If those aren't enough nicknames of which you should be aware, there are 4 types of bacteria which are commonly referred to by an acronym describing their pattern of antibiotic resistance. Methicillin-sensitive Staph aureus is called MSSA. Methicillin-resistant Staph aureus is MRSA. Vancomycin-resistant Enterococcus is VRE. And any member of the Enterobacteriaceae family that produces an antibiotic inactivating enzyme called extended spectrum beta lactamase is often called ESBL. That concludes this lecture on the classification of bacteria. I hope you found it interesting and useful. The next lecture will discuss acute diseases which are commonly mediated by bacteria, including the specific microbial etiologies of each.